Respected resource person of today's program, respected Dean of Life Sciences, Head of the Department, and faculty colleagues, and dear participants, a pleasant morning to one and all gathered here. I am very much happy to associate myself as a felicitator in the 3-day virtual training program on microalgae organized by the Department of Botany. It gives immense pleasure to me to take part in the virtual training program organized by the Botany Department for the betterment of our students as well as the outside participants. I hope that this kind of programs will continue in the days to come for the betterment of the botany department students as well as lecturers. At the junction of At the same time, I would like to thank the resource person for taking part in our college program. Once again, I wish all the very best to all the participants and uh, congratulate the organizers once again. Thank you. Thank you very much.
Of this session, Dr. R. Anburaj, Associate Professor, PG and Research Department of Microbiology, Vivekananda College of Arts and Sciences, Kobiman, Namakkal. He did his master's degree, MPhil, and doctoral degree in marine biology at Annamalai University, Chidambaram. He has 10 years of teaching experience in various university and institutions. He upgraded himself by completing 5 diploma courses and 42 certificate courses from Imperial IIT Chennai and from various reputed institutions like Alison, Ireland, University of Cambridge, UK, Monash University, Australia, Public Health, England, the University of Newcastle, Australia, Coventry University, UK, Griffith University, Australia, St. George University, West Indies. During his tenure, he held various positions like coordinator for NAC, nodal officer, and placement officer. He has research experience as JRF in international DEF projects sponsored by Gulf of Mannar Biosphere Reserve Trust during 2008 to 9. Worked as a junior research fellow and senior research fellow in a central government project sponsored by Ministry of Environment and Forest, New Delhi during 2009 to 12. He guided totally 19 UG and PG students. He was honored with various awards and medals like Best Senior Faculty Award by National Faculty Awards, Nobel Research Academy, Puducherry on 2021. Distinguished Scientist Award by International Research Awards, elsewhere Jayawada on 2020. Best Senior Scientist Award by Nobel Research Academy, Puducherry, India on 2020. Hargobind Khurana Best Young Scientist Award by both Science Societies, sponsored by Tamil Nadu Scientific Research Organization, Tamil Nadu, and on 2020. He is the fellow of both Science Society, TNSRO, Tamil Nadu, on 2018, and that fellow by Leibniz University, Hanover, Germany, 2009. He is the life member of various professional bodies from 2019. He is the reviewer in International Journal of Creative Research Thoughts and International Journal of Applied Agricultural Sciences. He got a patent for the application of marine-based minerals as food supplements in the year 2020. He proved himself as a successful researcher by publishing 15 research papers in peer-reviewed national and international journals. He has also published four book chapters. In his career, he participated and presented papers in various national and international seminars, conferences, symposium, FDP, and in-plant training programs. He delivered his expertise as invited speaker in Vivekananda College of Arts and Sciences, Koveman, Namakkal, and Srinivas College of Arts and Science, Perambalur. He has organized 11 seminars, symposium, conferences, and workshops. With this brief introduction, we welcome you for kindly being the resource person for this session. He will be sharing with us his expert opinion on recent trends in microalgae. We fully appreciate your finding the time to be with us despite your tight schedule. I once again welcome you, sir. Thank you. Now the session is yours, sir.
Thank you so much for your nice introduction, ma'am. Thank you so much, ma'am. Ma'am, it is audible?
Sir,
Okay.
audible, sir. Sir, audible, sir. Sir, audible, sir.
Madam, slide is visible?
Yes, sir. Slide visible, sir. Yes, sir. Slide
Shall we
visible,
start?
sir. Yes, sir. Slide visible, sir. Yes, sir. Sir, full screen change பண்ணுங்க, sir. Full screen change பண்ணுங்க, sir. Full screen change பண்ணுங்க, sir. Full screen.
Ma'am, now it is okay?
No, sir. No, sir.
Now, madam? Hello?
Sir. 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 Sir, full screen change ಆಗಲ್ಲ, sir.
Change ಆಗಲಿಲ್ಲ
Change
ಏನಾಗಿದೆ
ಆಗಲ್ಲ, sir.
ಕಾಣಿಸ್ತಿದೆ? Madam, right now?
No, sir. No, sir. No, sir. No, sir.
ஹலோ
சார்
மேம் இப்போ ஃபுல் ஸ்க்ரீன் தெரியுதுங்களா? ஹலோ
சார் ஸ்க்ரீன் பிளாங்கா இருக்கு சார்.
மேம் இன்னொரு லிங்க்ல வராது, கொஞ்சம் அட்மிட் கொடுங்க.
ஓகே சார். So now it's okay, sir.
Ma'am, shall we start?
Yes, sir. Start பண்ணுங்க.
Okay, fine. Good morning, friends. Today we are going to discuss about recent trends in microalgae. So first of all, I would like to thank the Steameth our management, and personally I would like to thank the principal of Steameth our college, and the vice principal of Steameth our college, and dean of science Steameth our college, and the head of the department, and my bosom from organization secretary Dr. Muruganandan sir, and colleagues. I would like to thank in my personally. Okay, shall we go to in detail manner? Okay. Today we are going to discuss about recent trends in microalgae. Before entering to the topic, today we are going to discuss about what are the things. This is our overview. Today we are going to discuss about the general introduction about the microalgae, then growth of microalgae, processing of microalgae for a commercial product, and microalgae applications, and emerging technology. What are the technology we have used? So generally, microalgae is microscopic organism found in both seawater and freshwater. They can be classified as eukaryotic and prokaryotic. Eukaryotic means it's having a few nucleus. The eukaryotic microorganism. The best example for chlorophyta. It is generally called as green alga. Prokaryotic means it's having a false nucleus, uh, and it's also having some immature nucleus. So that kind of microorganism comes under the prokaryotic category. So the best example is cyanobacteria. It is otherwise called as blue-green alga or PGA or cyanophyta. So for 25,000 species already isolated and identified. This microorganism performed photosynthesis process, which is important natural mechanism to reduce the atmosphere carbon dioxide concentrations. So based on this criteria, nowadays we have mainly focused on the carbon dioxide emission, how to control the global warming, how to control the greenhouse gases effective. So based on that, we have focused on many research. At the time, we have mainly fixed to the microalgae. It is suitable for this kind of process. So most of the eminent scientists or global level, we have mainly focused on the microalgae. So it's having a lot of applications. It's having a simple biomass productions. It will use it as a food and feed to the some valuable byproduct, especially for ecological applications. So why algae is photosynthetic microorganisms means that grow in wide range of aquatic habitats. It includes lake, ponds, rivers, ocean, and even wastewater. So that's why these organisms, photosynthetic microorganisms. Another one peculiar character it is having, they can tolerate a wide range of temperature, salinity, and pH value. So based on this category, they have classified eukaryotic and prokaryotic microorganisms. Nowadays, biotechnological aspect they are mainly focused on the microalgae. So now it is a budding stage of biotechnology field. So it is a young branch of algal biotechnology. The microalgal biomass market has been around 5,000 tons per year, especially for dry matter. It means biomass, and generate a turnover of US dollar around 1.25 into 109 per year. So the biodiversity of microalgae enormous and represents and almost untapped resources. It has been established that between 2 lakhs and several million species exist compared with 2 lakh 50,000 species of higher plants. Then growth of microalgae. So generally, microalgae growth it is classified as a three different way, namely for autotrophy, heterotrophic, and mixotrophy. What do you mean by autotrophic? It means microalgae produce needed organic matter that energy by using carbon dioxide considered as a carbon source and sunlight as a energy source. This group of microalgae comes under the autotrophic group. In heterotrophic group, it's having organic compounds are used to both energy and carbon sources. So this organisms comes under the heterotrophic groups. What do you mean by mixotrophic? It means metabolism consists two kinds of stages. The first one is heterotrophic. In this stage, the organic carbon considered as a high level. The second stage is autotrophic. In this stage, the photosynthesis process will easily occur. The organic carbon content is low. So this characteristics having the particular microorganism, that organism comes under the mixotrophic category. The microalgae classes mainly for four category. The first one is Cyanophyceae. It is otherwise called as blue-green algae or PGA or Cyanophyta. The second class is Chlorophyceae. It's nothing but a green algae. And third one for the Porphyridiphyceae, it is red algae. And the fourth category is Vesclophyraceae. It's include diatoms also. The Cyanophyceae having the many number of species. Among that, the one particular species can have a lot of biological activities. That species, namely for Arthrospiroplantanes, these microorganisms having phycocyanin pigments. That pigments can have a lot of biological activities, especially for anti-tumor, antioxidant, antibacterial, and anti-inflammation activity. So the best example for the Arthrospira plantanesis. The next category is Chlorophyceae. It is comes under the green algae category. It's having three potential species such as Chlorella, Hematococcus, Dunanella. So Chlorella species having a glucan and peptide bonds, especially for beta glucan and peptide bonds. That bioactive compounds is majorly responsible for some of the biological activities, especially for immune system stimulator, antioxidant, and blood pressure is reduced. The best example is Chlorella vulgaris. The second species is Hematococcus. This organism having astatinin pigments. That pigments is mainly responsible for some kinds of biological activities, especially for antioxidant, anti-inflammation, and anti-cancer activity. The best example for Hematococcus pulvalis. The third species is Dunella salina. It's having beta-carotene pigments. That pigments is mainly responsible for antioxidant and eye protections and anti-cancer activity. So the best example for Dunella salina, it's having a lot of biological activity. And the third category is Sporophyridaceae. It is comes under the red algae category. The best example were four-feeding species. It's having a polyunsaturated fatty acid. At the same time, it's having a phycoerythrin pigments. These two bioactive compounds are majorly responsible for antiviral, especially for herpes simplex antivirus and antibacterial activity, antioxidant activity, and immunostimulatory activity. The best example for Formedium porphyrium species. The fourth category is Bacillorosphaceae, includes dinoflagellates and diatoms. The best example for Photodialecum trichotinum, it's having a EP exopolysaccharide and phycoxanthin pigments. These two compounds are majorly involved for the improving the insulin productions, anti-cancer activity, anti-inflammatory activity, and cardiac vessel productions. Then what are the functions of the first microorganism? So we select the potential stains means it will easily we will create some bioactive compounds or bioactive products from particular microorganisms. At the same time, we can mainly focus on the cultivation conditions. And third step is harvesting and cell disturbance. And the fourth one is industrial applications. So first category is microalgae selections. These selections mainly based on the cultural conditions. At the same time, it will based on the bioactivity. For example, the cultural cultivations means we have followed two methods: open system for microalgal cultivation. At the same time, we can follow the closed system microalgal cultivation also we have followed. What is the major difference between the open system and the closed system? It means the open system having a good gas exchange with the atmosphere, easy to operate and scale up, cooling by evaporations, can utilize the sunlight as energy source, and as atmospheric air as considered as a carbon dioxide, it will consider as a carbon source. It's having a low installation cost and low energy consumption, but a high chance of contaminations. So this open system microalgae cultivation, we can face this kind of problem. In case we have cultivated closed system for microalgae cultivation, what would be the reason? The first point is good control on cultivation parameters. At the same time, it is high productivity one. To reduce the contamination risk, less carbon dioxide losses, and reproducible cultivation conditions. But at the same time, the cost of it is high. Cooling system is required, and scale up is very difficult. So these are all the major difference between the open system and closed system of microalgae cultivations. So this picture showing some diagrammatic. The first model is open system methods. We have followed maximum these three methods. The first one is raceway bond, and the second one is circular bond, and the third one is unstirred bond. In case of the closed system, we have mainly focused on the four methods. The first one is plate rector and column reactor and annular reactor, and final one is tubular reactor. The open burn system having the pedal wheel, and this system, the depth of the range is 15 to 25 degrees Celsius, especially for the raceway burn. In case we can cultivate the circular bonds, means the center axis having the agitator, the depth of the bond 30 to 70 centimeter in depth. In case of we can using for the unstirred bonds, means the bond area having the 50 centimeter depth range. Then what are the culture conditions? So generally microorganism will grow, means they will need carbon as well as the nitrogen source, or either carbon or nitrogen. This is the general mechanism of microorganisms. So based on these keywords, we can mainly focus on the culture conditions. So first thing is it is a photosynthetic microorganism, so it will need light source. So this is the energy source. What are the energy source we have utilized? Sorry, we can provide for the microorganism. And second category, the what are the carbon sources we have provided to the microalgae. And third one is light availability. And the fourth one, the based on this key indicator, the metabolism activity how will occur in the microalgae. The photoautotrophic means the energy source we can using provide only for light. The inorganic elements we can using for the carbon source, and the light availability we can using for the obligatory status. In heterotrophic group of microalgae means we can provide for the organic nutrients, that is considered as energy sources. The organic matter is considered as a carbon source, and light availability is required. Sometimes some group of microorganisms no requirements for the light availability. Then third category is photoheterotrophic microorganisms. This group of microorganisms it's need a light source. At the same time, we will provide some organic matters is considered as a carbon source, and light availability it is obligatory strategy. Then fourth one is mixotrophic microorganism. It means it will need light. At the same time, it will consider as a inorganic matters is considered as a carbon source. And third one is light availability. It is no obligatory. So these are all the some cultural conditions regarding microalgal cultivations methods. So once we can cultivate the microorganisms, especially for the open or closed system microalgal cultivation system, we can mainly follow some important characteristics. The first one is carbon source and nitrogen source, and optimum pH range and optimum temperature range, and it is a photosynthetic microorganism, so it will need the light source. So the light intensity it is mainly occur, and we can cultivate the cultivation method either open or closed system method. So this is the cultivation methods of microalgae. These methods we can mainly followed by open board methods. So directly we can using with the sunlight, so it will easily absorb the atmosphere carbon dioxide. So it comes under the photobioreactive category. So this group of microorganisms, once we can harvested, we can apply for the some product. So what are the steps we can follow? The first one is once we can collect it for the biomass means after collecting the biomass we can follow the three kinds of mechanisms. The first one is physical methods. The physical methods we can mainly focus on the centrifugation technique, sedimentation technique, and the filtration process or flotations. The second methods is chemical methods. These methods we can mainly use it for the flocculation technique, especially for the inorganic flocculants or organic or polyelectrolytic flocculants we can mainly use it. At the same time, some organisms we can mainly use it for the prefer for the electroflocculation techniques also. The third category is biological methods. These methods is mainly used for the bioflocculation technique or autoflocculation techniques. These methods we can mainly use for the flocculating algae, and at the same time we can increase the or decrease the pH range of particular microalgal. The fourth category is magnetic separations methods. So these are the general introduction about the algae and algal cultivations method. What are the key we can follow? So these are all the central informations. Now we are moving to the applications of microalgae. So microalgae having unique abilities to utilize them for industrial and environmental applications. It is used as a feed for animal and aquaculture. At the same time, it will used as a biofertilizer. It is mainly focused on the medicines field, especially for pharmaceutical industry, and we can derive for some cosmetic things from microalgae and environmental and other biotechnological applications. We can mainly focus on the microalgal culture. So, what are the applications having the microalgal means? It will play a vital role in mostly all the industry, especially for fuel productions, wastewater treatment, proteomics, and biomass productions, human nutrition and nutraceutical value, and biofertilizer and cosmetic things. So most probably all the industry we can mainly focus on the microalgal culture because it is having a lot of bioactive compounds. So based on this, we can mainly focus on the microalgal for all the industry in global wise. The best example for the promising features of microalgae, it means we can cultivate the microalgal culture and collect the biomass. So through the biomass, we can categorize the three kinds of product byproduct. The first one is direct usage. So directly we can using by the biomass microalgal culture we can using for the human food. At the same time, we can use it as a feed in animal and nutrient deficiency. That kind of people it will consider as a food supplements. So these are all the direct way you can using by the algal biomass. In case the algal biomass, it is majorly categorized into two byproduct. First one is byproduct and the second one is biofuels. So byproduct means it will synthesize a lot of polyunsaturated fatty acids, antioxidant activity, either enzymatic or non-enzymatic antioxidant activity, and it will synthesize a lot of pigments. So it will act as a coloring agents, and it's have a lot of vitamins. So it will lead to the vitamin deficiency patient. It will easily to recover that kind of peoples, and it will product from anti-cancer drugs because it's have a lot of bioactive compounds in microalgae. So they can mainly we can focus on the anti-cancer drug and antimicrobial drug also we can extract it from the microalgae. So this is the one part. The another part is biofuel productions. So the biofuel productions we can mainly focus on the three states. The first one is solid state biofuels, and the second thing is liquid. Third one is gas. So the solid state which means biochar. In the liquid state, we can mainly focus on the bioethanol or biodiesels. Sometimes we can use it for the vegetable oils. The third category is gases. It's nothing but biohydrogens or biosyngases. So this kind of these are the some promising features of microalgae. So why nowadays we can mainly focus on the byproduct as well as the biofuels because uh, algae having a lot of applications especially for wastewater treatment and atmosphere carbon dioxide mitigation techniques nowadays we can mainly use it. At the same time, we can derive a lot of byproduct especially for polysaccharides and lipids because lipids content is will mostly around 75 percentage of lipids it will occur in the microalgae. So easily we can extract the lipids and as well as we can extract the many kinds of pigments. So not only that pigments, it will having it is having also bioactive compounds. That bioactive compounds is mainly used for the anti-cancer and the antioxidant activity. So coming to the biofuels, it is the interest in microalgae because renewable and sustainable feedstock for biofuels productions. It is mainly newly focused in the terminologies biorefinery. So biofuel or a non-polluting one. At the same time, it is locally available and easily accessible one. At the same time, we can easily sustainable and reliable fuel obtained from the renewable sources. So biofuels we can derive from the microalgae means it will eco-friendly one. At the same time, it will non-toxic and strongly it will potential of globally carbon dioxide fixing mechanisms. So biofuel productions, microalgae mainly we can use. So most of the scientists they have mainly focused on the this kind of aspect, how to enhance the biofuel productions from microalgae. The next one is coupling of microalgae. It means microalgae we can cultivate in the wastewater. So the cultivation system having the two kinds of system, system. The first one is open bonds and the second one is photobioreactors. So this two system to remove the nitrogen content as well as the phosphorus content. So after removing the nitrogen and phosphorus content, we can harvest and dewatering, especially for the dried biomass. That dried biomass we can usually use for the value added product. Sometimes we can go for the bio components, especially for carbohydrate in sugar profile. In case we can use them for the many proteins. Among the protein, we can extract for some amino acids profile. So it's having a lot of lipids. That lipids having the fatty acid profile. So these are the biocomponents aspect we can mainly focus. At the same time, we can mainly focus on biofuel productions. So this production is mainly focused on the biogas productions and biohydrogen and bioethanol and biodiesel. So these are the aspects we can mainly focus on the biofuel productions from microalgae. The next important thing is microalgae. It will play a vital role even in environment and agriculture aspect or pharmaceutical or cosmetic, whatever it may be. The most of the industry we can mainly focus on the microalgal byproduct. So first thing is animal nutrition. In the sense we have mainly focused on the it will use as a source of protein and carbohydrate, vitamins and other components. So it is mainly used as a animal feed. The second category is food additives. So it's got a lot of pigments, especially for beta carotene and astatinin and polyunsaturated fatty acid. So it's having a omega-3 fatty acids. So these are all the pigments we can mainly applicable for the some nutraceutical and food additives. Then third category is cosmetic and pharmaceutical industry because it is having a beta carotene pigments and astatinin pigments and polyunsaturated fatty acids. So these are all the components we can mainly using for the cosmetic productions as well as the pharmaceutical industry. And coming to the bioenergy aspect, it will give the bulk oil feedback. So it will consider as a fuel and the biodiesels are the feedstock converted to the bioethanol or biogases or biochar. In agriculture basis, we can mainly use by the biofertilizer because it's having a nitrogen and the phosphorus sources in very rich, so it will easily considered as a biofertilizer in agriculture field. Then another category is wastewater treatment. It means this category the removal of nutrients from wastewater. Then another category for carbon dioxide captures from atmosphere or fuel gases emission. Microorganisms easily absorb the carbon dioxide captures. So we can using or we can working in microalgae means what are the key indicator you will get it. The first thing is you will be become a good researcher or you will become a good industrialist or you will become a good entrepreneur. So this three category you can easily experience while working in microalgae. At the same time, you have a lot of changes while using the some technology. You can face the some challenges also. The first one is high installation and operations cost. So we can nowadays we can using a lot of technology. So that technology is having a cost effect is very high. So the operation cost is very high. So this is the one big challenges for many industrial peoples. And second category is culture conditions. So culture conditions, the seasonal changes or physicochemical parameter we cannot appropriate methods we cannot follow means easily it will contaminated. So the culture conditions controlling system is another one big challenge. Then third category is microbial contaminations of medium. So generally microorganisms it will spread everywhere. So it will easily contaminate the that particular medium. So we can prefer for the specific medium for specific organisms. So that is known as selective media. At the same time, we can using with some peculiar medium for particular microorganisms. Then another one change is unbalanced supply for light and weather conditions because microalgae having a DL methods around 14 hours for light source and 8 hours for dark light. So DL methods it will easily adapted for some light source. So once we cannot provide the particular lux of light means we cannot cultivate the particular microorganism because it is photosynthetic at the same time it is autotrophic microorganisms. So the light source is failure means the cultivation methods also failure. What are the emerging technology we can using for the development of microalgal cultivations? So recent technologicals we can mainly develop seek to address these barriers. The first one is genetic engineering of microalgae. Genetic engineering of microalgae means the microalgae are rapidly emerging resources for genetic engineering, which means which enable to germinate the availability of sequencing genome across the multiple algal lines. The genetic engineering of microalgae can help to overcome the limitations of metabolic capacity and a higher accumulation of biomolecules that improve the economic facilities for the product process. In general, the genetic engineering we can mainly focus on the two bases. The first one is resource generation and enrichment. The second category is strain development and resource refinement. What is the difference between the resource generation and enrichment means? And the first thing is the screening of promising algal strains. So, which microorganism it potential strains for the particular byproduct? First, we can screening. This is the one of the promising source of algal strain. And the second thing is it is highly quality genomic scale because once we can find out the potential microorganism means we can cultivate the pure form and pure culture technique we can follow. Then next we are move on to the omics generations. So omic generation we can mainly focus on the some genobiotic metabolic models. So that model it is considered as a pure culture or it is contaminated one. We can easily find out. So once we you can find out that it is comes under the peer culture means directly we can go for the high quality genome scale and metabolic reconstruction process. So this all the process comes under the resource generations and enrichments. The second category is strain development and resource refinement. It means to regulate the what are the genes is present and what are the elements is available. That two things we can regulate the particular microorganisms and we can mainly focus on the gene editing process. At the same time we can create some models especially for the omics database and we can infratonic fluxomic studies also we can mainly focus on the strain development process. And finally we got the product development especially for the scale up stage. So this is the resource generation and enrichment. So this process we can mainly focus on the many extensive research work we are working in a microalgal because it will generate a high quality database. At the same time, the genomic and transcriptomic data, this model mainly provided for understanding of biosynthetic pathway. And third category is improved information. It comes under the genomics and proteomics. Especially, we can mainly focus on the metabolic activity. So, based on this metabolic activity, we can mainly focus on the byproduct from microalgae. And the metabolic data we can once we can implement it from the novel microbial strains or novel microbial isolates from particular genomic or transcriptase informations. Then second category is strain development and resource refinement. So in this category, this leads from some metabolic models, and it will lead to some out technologies. Uh, Best example for genome editing and high throughput variant selections. So it is mainly focused or mainly used the strain development methods, especially for microalgal culture. And the second category is metabolic flux. So it means the mutations of implements, especially for evolutions of some microalgae. So based on this evolutions methods, we can optimize the or rearrangement the uh, genomics from particular microalgae. And third category is flux balance. It means it will mainly focus on the biomass productions or enhancement productions of a particular byproduct or desired product. That kind of mechanism we can mainly focus on the flux balance shift method. So apart from that, the moreover the information is obtained from fine-tuned modeling or genomic editing experiments. It will create the further discoveries. So, genetic engineering, we can mainly focus on some genetic tools, especially for bioinformatics tools and transformations, vectors and promoters and transcription factors. At the finally, we can using for the mutagenesis also. So these are the tools we can mainly focus on the microalgal culture, especially for the biotechnology aspects. These tools we can using, and we can derive a lot of byproduct, especially for the biomanufacturing. The best product for food and feed, and another one is biofuel, and third category is recycling process. We can derive a lot of biomaterials, and final thing is medicines. So these are all the byproduct is mainly focused on the recombinant DNA technology or genetic engineering technology and biotechnological aspects. Next field from microalgae enter to the nanotechnology. So nowadays the nanotechnology one of the boom field of applicable life science or applicable bioscience. So nanotechnology is microalgae of a different way because it is exploiting the biosynthesis of nanomaterials, either molecular or cellular level. So this categories comes under the exploitations of microalgae. The second thing is it is a versatile process of microalgae because it's having a nanobiotechnology. Not only for nanobiotechnology, other microorganisms also we can using by the nanotechnology field. It is one of among the interesting advantage. So algal culture we can using by the nanotechnology means mainly we can focus on the metal nanoparticles. So metal nanoparticles from microalgae, it is one kinds of green approaches for synthesis and characterization of biological activities. So once we can correct the biomass of microalgae, then extract the biomass, then allow to the ionic solution metallic components, and the particular microorganism can able to synthesize the metal in nanoparticle. It is capable or incapable. We can find out. In case it is synthesis the metal nanoparticles means we can come to finalize the UV spectrophotometer and FTR spectrophotometer and some more time and XRD. Sometimes we can go for the AFM also. In case our microalgae having synthesis the biological activity of metal nanoparticles means we can mainly we can focus or mainly we can use it for the antibacterial and antifungal agents. Then recent category is Internet of Things. It's shortly called as LOT. It's help real-time monitoring of microalgae, especially for the biorefinery process. It will easily predict the what are the parameters we can follow for the biorefinery process. So it is a one kind of advantage. And second thing is it is collect from some sufficient data collections. It will make the smart predictions and decisions. So we have a lot of database. Based on this database, we can collect the lot of informations by using the lot of technology, especially for the biotechnological tools. Among these tools, we can conclude this microalgae easily predicted or non-predicted due to the based on the bioactive compounds. And third category is biorefinery process. So it is a automation process. So it is easily promoted as a biorefinery process. At the same time, the cost effect is low. At the efficiency level is high. So this is one of the major advantage of Internet of Things. And how to cultivate the Internet of Things means we can follow up with some applications methods. So it is automatic control point scanning microalgal cultivations. It is easily monitoring and it is easily manipulating the microalgal cultivations parameters, especially for pH, temperature, and blah blah blah. And next, microalgal productivity, it is mainly optimized based on the physicochemical parameters, pH, carbon source, nitrogen source, and pH or temperature, whatever it may be. Then coming to the identification of toxic algal species, we can easily screening and target microalgal species. At the same time, we can easily classify and microalgal species is viability detections or it is microalgal cells. So this is the one of the best method in nowadays. So first thing we can go for the real-time monitoring. It means data collections. So what are the microalgal species we can using? First you can find out or you can screen the which microorganisms it is potential for it will create some byproduct. First you can screen that particular microorganisms. So that screening process you should collect some data collections. So that based on this data collections we can go for the next analysis that is called as model inputs and data transmission process. So this process we can mainly focus on the machinery learning. It means it will mainly focus on the stimulations of modeling. So what are the growth factors we can use? What are the physicochemical parameters we can use? Based on these parameters, it will stimulate and it will frame the some modeling scale. So based on this modeling scale, we can go for the model output. That model output is mainly focused on the machinery learning and the automation process. So this process mainly for the DSS and optimization control points. And fifth category, the microalgal planets. We can once we can develop. So microalgal planets once we can develop means we can go for the upstream and downstream process. So we can using by the bioreactor or biofermentation process. So based on this biofermentation process, we can go for the many kinds of byproduct from microalgae. So upstream process means mainly we can go for the microalgal cultivation, especially for the biomass productions. In case of downstream process, we can mainly focus on the harvesting at the same time drying process and separations, screening classifications and biofuels, especially for the high value byproducts. It is all comes under the downstream process. So this is the summary of today lectures. So first thing is we can go for the carbon dioxide mitigations process. And next thing is the microalgae. It is majorly used for the wastewater treatment, especially for inorganic nutrients, contaminated water content. And third category is it is used is a lot of industry, especially for food industry and agriculture basis. We can mainly use it for the fertilizer. In case of health in means it will use it as a health product, and pharmaceuticals also we can use it for the some byproduct. And another output is it is microalgae derived from a lot of byproduct, especially for lipids, sugars, and protein contents. So based on these contents, we can go for the three kinds of main product. The first one is based on the biochemical conservation. We can derive or we can produce the bioalcohol or biodiesels or biogas. In case of direct conversion method, we can mainly go for the oil consumptions and animal food and fertilizer process. And third categories is thermochemical conservation. So this method we can mainly focus on the biocrude or biodiesels or biofuel synthesis. So microalgae industry is a large scale industry and provide a lot of alternative and sustainable byproduct that shows high interdisciplinary work of microalgae biomass that could interfere in more than one sectors. Using the advantage of new technologies in the microalgae industry would be great benefit to environment and to the product quality, which would be assembly reflect a better living standard of society, because it is a eco-friendly one. At the same time, it is a non-toxic byproduct. So human as well as the environment both are green and safe. So we can go for the many byproduct from the microalgae. So it will create some awareness program, especially for society based awareness program we can create. So I would like to thank STEAM and our Arts and Science College management, magnanimous management peoples, and I would like to thank Dr. J. Radhika, Principal Ma'am, and I would like to thank Dr. Pichamani, Vice Principal Sir, and I would like to thank Dr. Jodi, Dean of Life Science, and I would like to thank Dr. Ramesh, Head of the Department of Botany, and I would like to thank my bosom from Dr. Muruganadam, Assistant Professor of Botany, and other faculty members. Personally, I would like to thank I would like to thank my fruitful thanks to all of them. Okay, friends, if you have any doubts, kindly just ask to me. If there is no punishment for any asking questions. So now open for discussion. You will start.
Thank you, sir, for your inspiring talk on recent trends in microalgae. So there are few questions in YouTube channel, sir. During the cultivation of microalgae, how can we avoid contaminations? Sir?
Yes, ma'am. Yes, ma'am. So the cultivations method mainly we can follow the some physical chemical parameters. So each and every organism having the some particular pH or particular optimum temperature range. So based on this criteria, we can mainly focus on the which microorganisms are having the what kind of pH and what kind of temperatures. First, we can standardize the small scale sector. If you standardize a small scale sector means we can go for the large scale industry means we can easily console or we can easily overlap this kind of problem.
Thank you, sir. Next question, sir.
Tell me.
Any other common media for culturing all kinds of microalgae?
No, it is differ from organism because microalgae present in aquatic environment, even terrestrial as well as the marine environment. So it is differ from different kinds of aquatic. So it is not used for any common medium. In case of marine aspect, we can using for the some selective medium. In terrestrial microalgae, we can using for the some selective medium. So it is very difficult to use the both of the common medium.
So next question, sir. Can you please explain about the cultivation of Spirogera?
Actually, cultivation of spirogyra, it is very lengthy process. If you have any documentation, if you need, you just write my mail, dr.anbraj@vikas.org. I will send through the specific microorganism for what are the method we can follow or what are the techniques we can refer. I will send back to you.
Thank you, sir.
Thank you.
Next question, sir. Is there any specific tool for identification of microalgae?
Oh, no, ma'am. Actually, some marine organisms we can mainly focus on the identification methods. Some kinds of dye they are used. I think actinium orange. So, particular microalgal groups we can using where the actinium algae. I think Proclococcus domain. So that particular group of microorganisms we can mainly focus on the actinium orange dye. So it is differ from order or it is differ from classes of microalgae. So there is no common method for identifications or isolations of microalgae. Still now it is no one is find out. I think so.
So thank you, sir. Next question, sir. What are the algae which can be cultivated in mass production?
What are the algae?
Which can be cultivated in mass production, sir.
Oh, mass productions. So normally red algae we can prefer for the mass scale productions because rhodophyta species having a lot of bioactive compounds. The most of the industry people or most of the entrepreneur mainly cultivated the red algae species. So this is one of the mass scale production mainly their focus. Coming to the research aspect, we can mainly focus on the green algae, especially for chlorophyta. So this kind of species we can mainly focus on the research base. So depends upon the business or research, we can select the red algae or green algae.
Thank you, sir. Next question, sir. Why Nasdaq haven't a sexual reproduction?
Why now start?
Haven't next sexual reproduction, sir.
So sexual. Actually, it is absence of some chemical. I do not know. I forgot the name. That chemicals it is responsible for the reproduction system. That chemical is absence means it will not stimulate the particular reproductive system. So based on this criteria, this comes under the absence of reproduction of in Nasdaq.
Thank you, sir. Which will be the best technique for isolation of algae from soil?
Algae from soil sample.
Sir,
Once again, ma'am.
which will be the best technique for isolation of algae from soil?
Oh, soil sample.
Yes, sir, from soil sample.
It is depends upon the pH range. So, being a marine biologist, I working in the marine water. So, I can go for the Vignesgraphy medium, especially for water-bury techniques. In soil, exactly, I do not know. Sorry.
Okay, sir. Which is the best accurate technique of the species to identify the algae, whether morphological or molecular level?
A molecular level, we can easily go for the gene sequencing. In morphological, it is very difficult. So, the Bible of the morphological characteristics, Iyengar. So I think there is no new books or new manual. It is not available. I think so. Yesterday, Dr. Alumrugan saying any manual, recent manual apart from Iyengar.
Yes, sir.
Which one, ma'am?
Yes, sir. Sino? Cyanobacter, sir.
Cyanobacter. Oh, okay. So I'm not a expert from the morphological features. So I do not know.
Okay, sir. Next question, sir. Is there any specific medium for nitrogen fixing microalgae?
Yeah, nitrogen fixing microalgae. Which microalgae? Any specific microalgae or common?
Sir?
Ma'am, any
Microalgae, sir.
commonly they are asked.
Commonly they are asking, sir.
Okay. Nitrogen fixing process mainly for which means Vinagrosky or I think Vinagrosky medium is better based on my experience.
So next question, sir.
Tell me, ma'am.
Which is the best medium for spirulina cultivation?
PG11.
Okay. Any microalgae used in pollution remediation?
Yeah, a lot of microalgae use best example for chlorella. So organic matters it will mostly occur in the water source means the chlorella domination is more. Another one example for Azolla. So this organism is pollution indicator microalgae. Nowadays we can use.
Okay, sir. One last question, sir.
Okay.
Which microalgae contain rich in lipid?
Lipids.
Yes, sir.
Mm. So generally microalgae having 50 to 75% of lipid contents and which microorganism specifically means I tell you in marine aspect Synechococcus elongatus. So this organism have a lot of lipids. It will mainly using for the carbon dioxide sequestration process. Nowadays we can mainly use as a carbon sequestration process. In terrestrial, the lipid contents is which
Thank you, sir.
micro? Okay,
Thank
madam.
you, sir, for your inspiring talk on recent trends in microalgae. We would like to thank the Honorable Chief Guest, respected Principal, and our staff members who made this all possible. Thank you once again. Gratitude is the key to manifesting abundance. I would like to invite
Is the key
Dr.
to
J.
manifesting
Vanithamani,
abundance.
Assistant
I would
Professor,
like to invite
PG
Dr. J.
Department of
Vanithamani,
Botany,
Assistant Professor,
Srimad Andaman Arts and Science College,
Professor,
to express her
Department of
gratitude.
Botany, Srimad Andaman Arts and Science College, to express her gratitude.
Thank you.